Hi everyone, welcome to my talk. My name is Dudley and I am an Associate PhD student of the Centre of Excellence for Plant Success, and currently I'm based at Coffee. I started my PhD project this year, and my project focuses on the exploration of the molecular and physiological controls of flowering behaviour in Mungbean. So I too would just like to do an acknowledgement to country. I acknowledge the traditional owners and custodians of this lands we meet today and their contributions to Australia and society. So first I'll give you a bit of background on Mungbean if you're unfamiliar. So Mungbean, it's a high protein pulse crop which is grown in the subtropics of Australia, and it's quite an economically important crop for Australia. It's a main summer rotation crop, and it fits really nicely into the intercropping system with two main planting opportunities, either spring or summer. And it has a really short season, so it's got great flexibility for farmers and can make the most of advantageous rainfall periods. So as I mentioned, it's economically important for Australia, earning growers about $1,100 per tonne. And in fact, it has an export value of about $118 million. And about 90% of the Mungbean grain we produced is exported, as Australia is really well known in overseas markets for our high grain quality. So we've got quite advantageous place in the market at the moment. Also, pulses, as we all know, are really highly beneficial in agriculture because of their nitrogen fixing properties for soil improvement, and also having sustainable agricultural practices with intercropping methods. But Mungbean's actually quite a new crop in Australia. It's only been grown here since about the 1990s. And thus that means that there's been limited research into the molecular and physiological components of Mungbean grown in Australia. And we currently have quite limited genetic information available. But this really highlights that we've got some significant opportunities to expand upon the current knowledge and research and novel breeding methods which we might be using. So one of the main things for Mungbean is it's quite sensitive to abiotic stressors, and this is exacerbated by the long asynchronous flowering window of this plant. And this complicates harvest strategies and management practices due to the varying rates of maturity. So I've got a photo here on the right which displays this. We can see that we've got pods senescing and filling out, while we still have some new green ones developing and this continual flush of flowers. So this asynchrony may have had some significant evolutionary benefits in terms of reproductive survival, where stress may have undermined the first seeds produced. So these multiple flushes can be quite advantageous. But synchronization could improve the source sink dynamics, because if you think about it, there's probably these compensatory mechanisms if the plant is simultaneously producing vegetation, flowers and pods, and potentially even new roots. And this is probably going to have impacts on potential yield. So a greater synchrony in the flowering period and the reduction of the window will reduce the time which seeds are vulnerable to stress. So it's quite worthwhile for investigations for farmers. So as I mentioned, the difficulties of this flowering window on harvesting system and agricultural management differ depending on the different scenarios which people use. And in Australia for management, we currently use mechanically harvested systems. And the asynchrony of this flowering window increases difficulties for farmers deciding when to spray out the field for greatest yield benefit. And it can also result in the difficulties with desiccants decided to be used, because obviously there's limitations on desiccants that can be used depending on health and safety regulations if the pods are at varying stages of development. And this indecision for growers can make the crop more vulnerable to stresses in the environment, which we actually experienced this year with our own field trial. We waited too long and ended up losing all of our yield in the rain, but that's okay. It just really highlighted the scenario for us. And then other countries use hand harvesting systems. So Southeast Asia is quite a big producer of Mungbean in the international market. And in countries such as Myanmar, they use these hand harvesting systems, which are quite labor intensive. And when we're having these multiple flushes of flowering, it has multiple harvesting events, which increase labor costs. And it also results in a higher risk of lodging and chattering. So with both of these methods, the indeterminate flowering window increases costs and results in lower yields and quality for farmers. So how do I aim to identify what's going on and what is underpinning this asynchronous flowering window? Well, first I want to look at the mechanisms of flowering. So I actually have a little bit of a background in some of the fundamentals of flowering, and I want to look at how we all know that flowering in plants is quite a complex regulatory network, and it involves different environmental and endogenous signals. And I've got some of these signals on this figure here. These complex regulatory networks are integrated through some key integrated genes, such as FT and TFL1, which have antagonistic functions in flowering by activating and repressing these pathways respectively. And this control of the flowering pathway is through the determination of the vegetative or reproductive stages of the plants. And I think that these sort of genes involved in the integration of signals could be really useful targets for future experiments. And understanding the flowering time and the regulatory pathways which underpin this really critical development stage are fundamental to the plant's success and improvements in agricultural outputs. So we've begun to dissect this pathway in fundamental plant species, but it still warrants further investigations in a lot of our crops, especially mungbean. So as I mentioned previously, flowering is controlled by endogenous and environmental signals. And one of the most important environmental signals for flowering regulation is photoperiod, which is day length. So it's an important environmental cue that regulates flowering. And the transition from the reproductive phase is a key developmental decision for the plant because it sets a major change in the allocation of resources and ends the cycle of many different species. And currently, flowering behaviour in mungbean is quite diverse, and we've got varieties being classified as photoperiod sensitive, insensitive, determinate, or indeterminate. And currently, the next-gen model uses an older variety called King, which is what most of the photoperiod studies were based on. And then previously, before that, the model had no photoperiod sensitivity and was in fact just based on model. So we don't have any current photoperiod studies on any of the commercial Australian varieties. And this creates issues for expanding into potential production environments. Photoperiod sensitivity in mungbean really warrants further investigation, not only so we can understand what is regulating this flowering, but also the potential areas we can expand to for production. So as everyone here knows, our staple foods originate from crops, from ancient crops. So most modern crops can exhibit low genetic diversity compared to their wild relatives, from which they are domesticated because farmers have selected different varieties based on beneficial local adaptations. And this selection has created domesticated bottlenecks with limited genetic diversity. And to look forwards into advantageous agricultural development, sometimes we have to look back. So in mungbean, we have limited genetic variation for flowering time in most of the breeding programs, as plant varieties have largely been selected for disease resistance. And there's limited diversity in flowering time because we have quite a low genetic pool for this. And many of the physiological traits which have been selected for relate to disease resistance. So we've got really strong benefits in this pool for disease scenarios, but not in regards to abiotic stress conditions. And it's got a really narrow flowering window across most of the commercial varieties used in Australia today. So with modern plant breeding strategies, we can analyse different diverse populations by assessments of different genetic and phenotypic diversities and have a look at the available genetic resources and use it as a primary step for launching future research into projects for useful genes in crops. And so this is what my project mainly focuses on, is using these diverse populations and seeing if we can have a look at the variation in flowering time genetics. So first of all, I've been working with QUT and DAF's NAM population. They've developed a huge population, which is a multi-parent mapping strategy, where it's got multiple diverse donor parents. And in this, they used one of the main commercial Australian varieties called Crystal, and then some interspecific crosses with Australian native species. And I've used several parents from this panel to create a diverse panel for many of my future experiments. And these lines comprise of different species from different locations all around the world. So on this map here, we've got some lines from Pakistan, India, Bangladesh, Myanmar, Taiwan, Philippines, and even our natives from Australia. And it's quite diverse, and I'm hoping to look at a number of different aspects across this panel through the course of my PhD project. So these are some photos from the first experiments I've been conducting, and you can see here we have quite big differences in the morphology of these plants. We've got a commercial variety here, a wild Australian mungbean line, and then one of the breeding lines which originated from India. And there's not only differences in the flowering behaviour, we've got also differences in leaf morphology, with most mungbean having a trifoliate leaf pattern, but our Indian line has 5 leaves instead of 3. And then differences in the flowering window, which is one of the first things I've looked at. So through the course of my project, I aim to elucidate the physiological and genetic controls of flowering in Mungbean, and I'm doing this through different targeted molecular, genetic, and physiological strategies. And hopefully my results and information gathered through the course of my project will lead to optimized flowering behaviour and information for breeding programs. So the outputs of my project will hopefully contribute to pathways to accelerate Mungbean breeding programs, and this is through the use of analysing diverse populations for varying genetic resources to kickstart this. So with this diverse population, I'm looking at variation in different traits and behaviours, and hopefully being able to understand the physiological basis of flowering with these traits. Then using the information from this population, we'll do high throughput genotyping to identify gene targets and investigate orthologs, and then hopefully combine this with several field trials where we're using UAV methods for high throughput phenotyping to accelerate the different pathways. And hopefully this research could increase the genetic gain for Mungbean, improve the Mungbean AppSim model, and also improve prediction technology that we have for phenology and target production environments. So for my first experiment with this diverse parent panel, I'm conducting several photoperiod experiments where I'm growing these 31 parents under different photoperiod lengths. So I've got a neutral day, which consists of 12 hours, and short day experiments. And my short day, which is about 8 hours, has just finished up. So I've got the results in this PowerPoint today. And hopefully once I have identified different levels of variation in the parents of these panels, I will be further refining these experiments to examine a broad fitness landscape to understand how flowering behaviour is influenced by different abiotic stress conditions, such as water stress or different heat stress environments. So I've got the results from my first experiment, which was the panel being grown under short day conditions. And this graph does seem a bit overwhelming because there are lots of lines included. So don't worry, I will break it down a little bit with some of our most interesting lines. But traits that I was trying to quantify, I've got a little example of a Mungbean flower here. I was looking at numbers of flowers, the flowering windows, days to first flower, and something that I noticed in this experiment, which I'd like to investigate further in my future experiments, is floral abortion rates. Because as you can see in this image, there's quite a few little buds around here, but sometimes we'll only have one pod. And this varies quite a lot amongst the different varieties as well. So it's something I'm interested in specifically when I'm looking at these source sink dynamics. So these graphs look at the nut flower number over time. And this first variety is Crystal, which is one of the main commercial varieties given to farmers by the National Mungbean Improvement Program. It was released in 2008, and it's quite highly regarded as one of the main commercial varieties due to this nice, large, shiny green seed, which is what makes Australia known for its high quality seed in the international market. So in my experiment of short days, it had these two flowering windows, with the first one resulting in the most number of flowers, and then the second window follows quite shortly after, which is quite interesting when I'm thinking about the dynamics and compensation between vegetative and reproductive tissues. Birkin is another line I included, which is a commercial variety. And again, it has these two flowering windows, although they are a little bit more separated in comparison to Crystal. And then for Solera 2, which is in the breeding material, we just have this larger single flowering window. So along our commercial varieties, we're already seeing a little bit of difference in the flowering behaviour, and it gets even more interesting when we have a look at our native variety. So here is one of my favourite lines. It's actually quite cute to look at. It's a lot more short growing than our commercial line with these tendrils, and the difference in flowering behaviour along the tendrils is quite interesting when looking at it on the main stem in our commercial varieties. And this flowering window just seems to be increasing through the whole duration of the experiment. It doesn't seem to stop and have the multiple flushes, so like the commercial varieties, and it has quite higher numbers of flowers. So as this experiment finished just last week, I'm also going to be dissecting traits like pod development and grains, so it'll produce some really interesting results. And then I've just finally included last two varieties because there's obviously a lot to get through with 32. I'm showing EJP2, which is in the breeding materials, and it has these much later and shorter flowering windows, and then CPI, which is an Indonesian originating line. And basically from these figures, I just want to really highlight the level of diversity that I've already seen in my panel. And I believe it's going to really present a lot of exciting opportunities in future investigation through the course of my PhD. So I'm currently in the midst of designing different projects where I can try and dissect the variation and diversity as much as I can. So another main part of my project, as I previously mentioned, is to try and understand the systems which regulate flowering in Mungbeans. So I want to do this through the exploration of flowering time genes and signaling pathways. So I want to create experiments to investigate the knowledge from model species on flowering time genes, and I want to understand what the functional controls of flowering are in Mungbean. So I'll be evaluating variation of sequences for flowering orthologs in these different Mungbean lines that I've got. And hopefully, once I've identified appropriate targets, I'm also working on a genome editing pipeline. We'll be able to conduct some functional studies using CRISPR-edited line to understand gene function. And then hopefully through some further analyses, I'll be able to develop methods to identify if we've got any key physiological traits which can be used to predict shifts in the flowering behaviour, such as looking at hormone or sugar levels in plant tissues. And hopefully this will be something that will also relate to another part of my pre-PhD project, which is where we're using multi-spec and hyperspec sensors to look at the shift in flowering and try and predict it in the field before we can actually score it. So this year, as I mentioned, I conducted a field trial with the QUT NAM population. It's got about 2,000 lines, and we had 3 different sites consisting of a total of 4,000 plots. And I scored these plots daily for traits such as days to first flower, 50% flower, 100%, and then end of flowering and flush and sweep after that. And also looking at things like determinacy and indeterminacy of the lines. So with the results of this trial, I have begun to conduct some GWAS analyses looking at these mentioned traits. And currently the developed genomic tools for Mungbean have been quite limited, but there's a recent reference genome for Mungbean and it provides new opportunities for these research, which is quite beneficial for my GWAS analyses. So this is the results of my GWAS just for the first trait we've been looking at, which is days to first flower. And we have identified 2 significant markers which haven't previously been found. And I'm currently investigating these markers for different flowering orthologs. So as I mentioned, I'm also working on a tissue culture editing pipeline. So I've been working in collaboration with the Ferguson lab here at UQ for the development of this tissue culture line. So hopefully with the production of this pipeline, I will be able to further investigate the results of my GWAS analysis, and also with the development of some future experiments looking at sequence variation in my panel. So hopefully these gene editing strategies will provide methods for me to look at modified flowering behaviour across my lines and also generate some mutants. The development of this pipeline will provide a really important resource to study the pleiotropic effects of flowering behaviour and looking at this on growth and development in Mungbean. So I also have been working with some of, as I mentioned, these UAV hyperspec and multi-spec technologies to see if we can predict floral induction pathways in the field environments. Essentially, what I want to do is to determine if it's possible to see flowering before you can physically see it by correlating multi-spec data with ground-based measurements, because it is quite time-intensive to take this data, and it's also just as time-intensive for these farmers in their management practice. Flowering actually initiates in Mungbean underneath the canopy. So in order to get this really refined data, farmers actually have to walk the fields daily to sort of see when this reproductive stage is initiating. So I hopefully aim, as one part of my PhD project, aim to develop these prediction models for flowering time using multi-spec sensors from UAV platforms. And I want to try and pair these predictions with some hypotheses, like looking at if I can use climate data, like figures such as growing degree days, to predict flowering in field environments, or also if these UAV platforms can obtain data shifts for physical characteristics and have a look at this as indicating flowering. So this is just something I'm hoping to, because I understand that my project is quite large at the moment, but from what I hear, this is a pretty standard thing for a PhD project, is just the first year being quite an investigative thing. So thank you very much. I would like to acknowledge all of my supervisors and people in my lab, which I've been working with and helping me very much. Thank you.